Okay, now we are ready to talk about the citric acid cycle. So this is kind of continuing on from the chapter about glycolysis. So remember that glycolysis is the main way that most cells convert glucose and other monosaccharides into energy by breaking it down into two molecules of pyruvate. So this is how pretty much all cells, including those anaerobic organisms like certain bacteria, for example, get their energy. And this is probably how most organisms got their energy until oxygen became common in the environment. Anaerobic organisms or cells in hypoxic environments, instead of once they make pyruvate, then further break down it into ethanol and carbon dioxide. This is again the process of fermentation. In animals, the pyruvate gets converted into lactate in the absence of oxygen. But in the presence of oxygen, or in aerobic conditions, then the pyruvate gets converted into one carbon gets removed to CO2, and it gets converted into acetyl-CoA. And then acetyl-CoA enters this process called the citric acid cycle, where it gets broken down the rest of the way. Basically, the rest of the energy available is extracted and converted into ATP or electron carriers, and then the carbons are converted to CO2 and water. So the energy availability of glycolysis is very high. This is why it probably evolved. But again, the full process of full oxidation of glucose requires oxygen, whereas glycolysis itself, you only get about 146 kilojoules per mole of usable energy from converting glucose to pyruvate. But then full oxidation gives you much more energy. This is again why it is such a universal method of energy extraction. But early on in Earth's history, there wasn't very much oxygen available in the atmosphere, so most, probably the earliest organisms did not use this process because they didn't have oxygen. So instead they would have had to have had some sort of anaerobic metabolic process. But eventually, when organisms like plants started producing oxygen as basically a waste product, it became much more available in the atmosphere. And this was about 2.5 billion years ago. And that presumably opened up the door to cells to fully oxidize glucose and get the most energy possible out of it. Again, glycolysis itself produces, if you remember, a net of 2 ATP molecules, but the full process of oxidation, which includes the citric acid cycle and electron transport chain, produces a lot more ATP. The other nice thing about this whole process, which we call cellular respiration, is that pyruvate can be produced through other pathways, including from lipids and some amino acids. And so, whereas glycolysis only is a way to extract energy from glucose, full cellular respiration allows you to get energy from other types of molecules also. And this kind of breaks the whole process down. So essentially, there's 3 stages to cellular respiration. The first is the production of acetyl-CoA. So for glycolysis, that means first taking glucose, converting it into pyruvate, which we've already talked about, and then converting pyruvate into acetyl-CoA. There's a completely different set of pathways for getting acetyl-CoA from amino acids and from fatty acids, but the end result is still this acetyl-CoA molecule, which then enters the citric acid cycle. So that's what this chapter deals with. But the citric acid cycle, along with some of the steps in glycolysis and some of the steps in amino acid and fatty acid catabolism, produce these electron carriers. That's what these little electrons are for. And so those electron carriers are basically a way of transferring chemical energy from these precursors down to eventually to a whole other set of reactions called the electron transfer chain. And that's where most of the ATP in most cells gets made. And that is the step also that requires oxygen. So again, this is why oxygen is necessary for this whole process, because the very last step requires 1 molecule of O2 in order to be basically the final electron acceptor. That is where we're at. So we're going to talk about the citric acid cycle in this chapter. So first of all, before you enter the citric acid cycle, the pyruvate gets converted into a molecule of acetyl-CoA, and that's carried out by an enzyme complex called the Pyruvate Dehydrogenase complex. So this complex consists of 3 different enzymes. It converts pyruvate into acetyl-CoA and then transfers an electron to 1 molecule of NADH. So it's kind of a complicated process. CoA is short for coenzyme A. This is actually a common acyl carrier for a lot of other metabolic reactions also. So this is 1 molecule of CoA. It has this reactive thiol group at the end, and that's actually where the acetyl group gets attached. So that's what the pyruvate dehydrogenase complex does. It just attaches that acyl group to this coenzyme A group. And this is the pyruvate dehydrogenase complex. Again, it's a strangely complicated process for just one relatively simple reaction. So again, you're starting with just this small molecule of pyruvate, and all you're really doing is attaching it to this bigger, more complicated molecule of acetyl-CoA, of coenzyme A. But it requires 3 different enzymes, several other cofactors, including a molecule of TPP and FAD, and it involves 3 enzymes. And these 3 enzymes work together very tightly. In fact, this is an example of something called substrate channeling, where the 3 enzymes, even though they each catalyze different reactions, the substrate of each reaction gets transferred directly from one enzyme to another. So rather than have the substrate kind of diffuse away from the first enzyme and move to the second, the substrate just sort of immediately transfers from one group to the next. But again, the end result is you end up with 1 molecule of acetyl-CoA. You do lose a carbon, so this is the first place where one of the carbons get lost from pyruvate, so you produce CO2. Remember that we started off with glucose if we go all the way back to glycolysis, and so that was a 6-carbon molecule, and glycolysis produces 2 3-carbon pyruvates, and now when we lose 1 to CO2, now we're down to just 2 carbons in the form of acetyl-CoA. And then that is a reaction that produces an electron. So this is basically oxidizing pyruvate to acetyl-CoA. And that electron eventually, again, through several reactions, gets transferred to 1 molecule of NAD plus to make NADH. And we'll talk about electron carriers more later. So then acetyl-CoA enters the citric acid cycle. And so the citric acid cycle is this pretty complex looking set of reactions. It's sometimes called the Krebs cycle because it was discovered by this guy named Hans Krebs. And it's sometimes also called the TCA cycle because TCA stands for tricarboxylic acid because the acetyl-CoA gets converted into citrate first, which is a 3-carboxylic acid molecule. And as we will see, the reason it's called a cycle is because the end product of all these reactions is also one of the first substrates. But several of these reactions produce produce NADH, again, as electron carriers. So the energy stored in this acetyl-CoA eventually gets transferred to NADH. There is a molecule of GTP that's going to be produced, which again can be used to make ATP or can be used in other reactions. And also FADH2 is another electron carrier. So it looks complicated, but it's really just 8 reactions. And what basically is going in are the 2 carbons of this acetyl group. They're going to end up as another molecule of this substance called oxaloacetate. But then 2 of the carbons from this oxaloacetate are going to leave as CO2 molecules. So it's a way of sort of recycling these molecules or these carbon atoms through this process. So the first reaction is converting acetyl-CoA into citrate. So the enzyme Citrate Synthase combines acetyl-CoA, basically takes the acetyl group off of coenzyme A and converts it back into just regular coenzyme A, and then it attaches that acetyl group to a molecule of oxaloacetate. So, so this is oxaloacetate over here, and then acetyl-CoA here combined to give you citrate. And you can see the 2 carbons of the acetyl-CoA are still here next to each other. So we'll kind of try to keep track of those as we go through. The next step is that citrate gets converted into isocitrate. So an enzyme called aconitase carries this out. Basically all that happens is this OH group over here gets moved to here. And again, that's carried out by an enzyme that actually removes a water molecule and then puts another one back on, so it's not actually the same oxygen, but that's basically what you're left with. So it's just an isomer of citric acid. But that sets up the next reaction, which is carried out by an enzyme called Isocitrate Dehydrogenase. And Isocitrate Dehydrogenase removes 2 hydrogens and transfers one to a molecule of either NAD plus or NADP plus, and then the other one is removed as a molecule or just as a proton, and then this carboxyl group is removed as a molecule of CO2. So this is the next carbon that gets lost from of course this carbon was not one of the carbons that got added. So remember that basically it was these 2 carbons that got added from pyruvate, so we're not losing those yet. These were carbons that were part of the original oxaloacetate, but again, this is a cycle, so that carbon was presumably part of a different pyruvate that passed through this cycle previously. But anyway, isocitrate gets converted into alpha-ketoglutarate, and again, now we've transferred an electron to a molecule of NAD+. So this is just to explain what that means. NAD stands for nicotinamide adenine dinucleotide. And this is just what the structure looks like. And different systems use either NAD or NADP. NADP is just a phosphorylated group where the phosphate is added here to this 2' carbon of the adenine group, and then the nicotinamide group up here is what actually gets the electrons, and so NAD plus is the oxidized version of this molecule. NADH is the reduced form. So this reaction produces a molecule of NADH and then a free proton. And then this NADH will be important later when we talk about the electron transport chain. Then that molecule of alpha-ketoglutarate goes through another oxidation where it gets converted into Succinate CoA. So we have another coenzyme A coming in. This enzyme alpha-ketoglutarate dehydrogenase basically takes off another carboxylic acid. Again, not one of the carbons that was added from pyruvate, but one that was part of the original oxaloacetate molecule. Another electron gets transferred and a hydrogen gets transferred to NADH. The hydrogen in this case came from the coenzyme A, not from the alpha-ketoglutarate. But in any case, you end up now with a molecule of Succinate CoA, and we've lost another carbon. So now we're down to, we've actually lost we're down to 4 carbons. We started off with 6. And so that's all the carbons that are going to be removed from this molecule. And then Succinate CoA gets converted into succinate, by Succinate synthetase, which just does the reverse of the reaction in its name. It takes off the coenzyme A and converts this molecule back into succinate. It also, in that process, produces a molecule of GTP. So GTP is just combining guanosine diphosphate with an inorganic phosphate. GTP itself is a way for the cell to use energy. It can either be used to make a molecule of ATP, or there are some reactions, some enzymes that use GTP as an energy source instead of ATP. They're actually called G proteins. And then the next step, that succinate gets converted into fumarate by an enzyme called Succinate Dehydrogenase, which does pretty much what it says it does. It takes two hydrogens off and converts that middle carbon-carbon bond into a double bond. It takes those hydrogens and attaches it to a molecule of FAD and makes FADH2. So this is, again, a process of reducing FAD to FADH2. So we're transferring another electron. So FAD is another example of one of these electron carriers. FAD stands for flavin adenine dinucleotide. So that means, and sometimes in some situations instead of FAD, just the flavin part, the flavin mononucleotide, is used as the electron carrier. But this is a molecule of FAD. This whole thing is a molecule of FAD. And then this part is the flavin mononucleotide. The important part is this isoalloxazine ring, because that's the part that gets reduced. So it can be reduced once to FADH semiquinone, and then reduced again to FADH2. So this is the fully reduced form of this molecule over here. So again, this is another electron carrier that will come up again when we talk about the electron transfer chain. The next step is that fumarate, then that double bond gets converted back into a single bond, but now we end up with another OH group. So this enzyme fumarase turns fumarate into malate, and then that malate gets converted through, gets oxidized again to oxaloacetate. So L-malate dehydrogenase just takes malate, transfers two hydrogens to one hydrogen to NAD+, another to just a free proton, and you end up with oxaloacetate as the final product. And again, oxaloacetate is the thing we started off with. So that's why we call this a cycle, because we used up a molecule of oxaloacetate at the beginning, and we end up with a molecule of oxaloacetate in the end. But again, it's important to point out that the two of the carbons of this oxaloacetate came from the pyruvate that we started off with at the very beginning. And so we didn't lose those carbons as CO2, but now when this oxaloacetate goes through the cycle again, it has a chance to lose those carbons the next time. So, and this kind of just summarizes everything without all the enzyme names in it, and gives you, again, kind of a balance sheet for what we started with, what we ended up with. So again, we start off with a molecule of acetyl-CoA. 3 NAD plus molecules get used up, and 1 FAD, 1 GDP, and 1 inorganic phosphate, and 2 water molecules go in, and we lose 2 carbons as CO2. We produce 3 NADH and 1 FADH2 molecule as electron carriers. GTP is made by combining GDP and inorganic phosphate. The coenzyme A comes back out, and we produce 3 protons. And that whole process, again, is the oxidation of 2 carbons from CO2. And in fact, those were the 2 carbons that were eventually, those would be the 2 carbons that came in as acetyl-CoA. The third carbon, remember, got lost when we converted pyruvate to acetyl-CoA. And so 2 pyruvates, again, contain the 6 carbons that came from our original glucose. So 1 molecule of glucose, eventually all those carbons get lost as CO2. In fact, this is why we we lose CO2 or exhale CO2 when we breathe. The CO2 that comes out of your lungs when you breathe out, those carbon atoms started off presumably at some point as some sort of organic molecule, either glucose or some other molecule that can be converted into pyruvate or acetyl-CoA, and then goes through the citric acid cycle. And then again, this whole process, the important part is the energy that was captured from the original acetyl-CoA gets used to make NADH and FADH2, and the energy there will be used to drive the electron transfer chain, and it produces some energy on its own. So that one molecule of GTP does get used as energy, either to make ATP or in some other reaction. Now, I don't necessarily want you to memorize all the steps here. Some of them are going to come up later when we talk about the electron transfer chain some more. But do kind of be familiar with the net reaction, what goes in, what comes out, and just keep those electron carriers in mind, because they're going to come up again later. Now, the last thing to say about citric acid cycle is that it's not just about making electron carriers. It's sort of a hub cycle of sorts, a hub reaction that is used in a lot of other processes. So a lot of these intermediates can be diverted into other pathways to make other biological molecules. Citrate, for example, can be used to make fatty acids and sterols. Alpha ketoglutarate can be converted into glutamate, which is one of the 20 amino acids that we use to make proteins, and then glutamate itself can get converted into other amino acids like glutamine, proline, and arginine. Glutamate also can be converted into the purines, which are, of course, going to be used for nucleic acids. And so there we go. And then Succinate CoA can be converted into porphyrins. So porphyrins are those big, complicated molecules that are eventually used to make things like heme and other molecules like that, which we'll talk about some examples of those actually in the next chapter too. And so that's where those come from, or can come from. And then oxaloacetate itself can be diverted to make other amino acids like aspartate and asparagine. And then asparagine can be converted into the pyrimidine. So those are the bases like thymine and cytosine that are used for nucleic acids. And then oxaloacetate also can be converted back into phosphoenol pyruvate, and that can be converted back into glucose. So that's one of the glucogenic pathways that we mentioned real briefly in the last chapter. And then that also can be converted into more amino acids. So serine, glycine, cysteine, phenylalanine, tyrosine, tryptophan, all of those can be made from phosphoenol pyruvate. So again, the citric acid cycle is not just an energy producing process. It also is a central way for the cell to make a lot of other stuff. All right, so that's all we're going to say about the citric acid cycle for now. Next time we'll talk about the electron transfer chain.